Okay, here we are, and on a crazy nice day outside, and we are starting. Let me just get a couple things organized here, and we'll go for it. All right, so this is where we're at. 2 days to basically cover the cell membrane chapter 14, and then maybe there'll just be a little bit next Tuesday, a week from today, a little bit of new material, but we should be in good shape. Some new material might come up if somebody asks a question and we dive into something that's from the book and let's explain it, or something we loop back to and want to add more examples to, like that. So remember that as before on the class meeting before an exam, I'll talk about new material. We'll finish the existing material for the exam and then talk about new material unless people have questions, And again, I think we'll be in good shape a week from today. Maybe cover just a few more, pick up a few more loose ends, but then we have a test a week from Thursday, our third test, and we know what it covers. The chapter on the nucleus, the chapter on protein synthesis and trafficking, and then this chapter on the cell membrane. So lots of really great cell biology that comes from our third section of our book, and goes back to the start of our course where I say, molecular biology and cell biology are actually two different topics, two different courses potentially, and I hope you can see in studying for this third exam, there's a somewhat different feel for it. I think it even becomes hopefully clearer to students that drawing stuff out in biology, spreading it out, making a great big space is really good for helping you, because learning is all about recall. And what is it that you recall? Do you recall my blabby voice and poor handwriting? Well, that's just sad, right? Like that. Or do you recall our textbook, and just the letters in a textbook are hard to see in your mind? Or do you recall yourself, creating, on a really nice day at the beach, creating some drawings like that? And so that's cell biology is what we're up to, really. And so we'll do that. And then our last week of class, we won't have new learning. I'll introduce some more new topics, wrap up our course. But we can also answer questions. So, we're getting close to where, do you have questions becomes more significant in our course than ever. And because we're going to be pulling it all together like that. And with regard to that, when it comes to like reviewing stuff, right? Students like the idea that I might plan a review, or have a study guide, and that's all I'm doing. And I know that sounds kind of offensive, but I've just distilled everything down as much as I can for all the class meetings. And I wouldn't know what to review, because I don't know what it is that a student would have found that we talked about that was confusing and needs to get looped back again, like that, okay? And so, be mindful of that. And then there's our, final exam. And so we're also making sure you're aware final exam is on Tuesday the 5th and 8 a.m. in our class. If you're not here right then, just come in and be quiet and get started. And then what are we doing this week? So this chapter 14 from our book about the cell membrane. This Thursday, this RNA processing assignment is done and is due. And that's the one where there's no reading required for it. It's just about the video, and it involves doing some drawings. And it is a really important one. We bring up some really important new stuff that fits into the pathway of regulating gene expression. And then third exam, and the third exam will look like our first and second exam. So just to show you, our whole third exam could be fit in this diagram, emphasizing that. So what's happening inside the nucleus? So for our assignment on Thursday, RNA processing. So we go back to saying, oh, that's happening inside the eukaryotic nucleus, and included in that video assignment is a process that's called RNA interference. So all those are on the pathway of gene expression. They're all steps that have the possibility of regulating gene expression. We could block gene expression by RNA interference. So, maybe the gene's being transcribed fine, the RNA gets processed fine, it gets out in the cytoplasm and it just gets destroyed, like that. And so all along that path, and then that chapter 11 was really focused on post-translational regulation, so after proteins are synthesized, and also how they are newly synthesized like that. And the cell membrane is the last one for us, and we'll be talking more about that. So just looping back to some of the stuff we talked about on protein synthesis in that chapter. We had that assignment that was due last week about describing these, and we have those 4 examples. And we would stick any other examples of protein synthesis that we have in our course on that list. We'd fit it on that list. Is it just made by free ribosomes like hemoglobin? It's just going to float around? Yeah, well, every transcription factor is like that. And then it's got to get into the nucleus, right? So a key step in regulating transcription factors post-translationally is you're going to let it into the nucleus or not, like that, right? So it all fits into this flow, and it's a good way for us to focus and talk about the whole cell in cell biology. So be aware of that I just in that assignment last week, I just distilled it down to these 4 classic examples, and other proteins, almost all other proteins would fit on those in those categories like that. And then we also talked, and I just want to follow up on this, we also talked about some post-translational modifications of newly synthesized proteins. So as they're newly synthesized and they're getting ready to do their job, what is happening there? Remember that there is additional post-translational regulation of a protein potentially at any time it's around. So MYC gets a lot of regulation in newly synthesized protein, or anytime it's around. It's like, okay, we got to regulate that. We got to ubiquitate it and destroy it, and things like that. So this last one I just wanted to follow up on, because I had a little glitchy issue in talking about it last time, so I got a figure to show you. And so it's RAS, and it's our last example, and I talked about it last Thursday. RAS gets synthesized in the rough ER. So it's an example of something that's going to have that signal sequence on it that's going to cause the ribosome to pause in the synthesizing of RAS, dock on the rough ER, and complete the synthesis of the RAS there. And that's because RAS is always been shown here to be anchored to the cell membrane. And that's because of a post-translational modification. So post-translational modifications means a covalent bond change to a protein, and it's modified. It's modified by having a lipid get added to it. And we don't need to know the name of the molecule it gets added because it has a really funny name. And where that gets added is when RAS gets synthesized in the rough ER, it gets added there. And so here's just an image of that idea that RAS is going to get synthesized here, and it's going to go through the Golgi, and it's going to end up on the inside of the cell membrane. And in our book, this is the glitch thing I had in our book. I showed you this image last time, and it's a zoom it in of the rough ER and the rough ER membrane. So this is from that chapter 11, and it's showing proteins that are in the rough ER and showing how they get positioned when they are synthesized. And I just grabbed this image when I was preparing the lecture and said, oh, this is a good example of RAS, because these guys are getting anchored. But then I was like, whoops, wrong side of the membrane. These guys are going to end up on the outside of the cell membrane. RAS ends up on the inside. And this is a good time to pause and see, visually that there's a big difference, again, and we talked about it, there's a big difference between the inside and the outside of the membrane, whatever the membrane is. There could be differences in the phospholipids, there could be differences in the orientation that membrane proteins are in that membrane, like that. So again, the lumen of the rough ER is going to become equivalent to the outside of the cell, because that's going to go through a vesicle and go through exocytosis, and everything that's here, whether it's a phospholipid or in the lumen or a membrane protein with that, that's going to be the outside of the cell. And RAS is on the inside, so this must be where the RAS gets synthesized. And it does get synthesized by a ribosome that's on the rough ER, but the RAS is just synthesized right there, and before the ribosome lets go of it so it could float away, because it, you know, in principle could, it gets that fatty acid chain added, that lipid chain added, so it's stuck there. And then again, the RAS is going to be there, and it's going to go through vesicles of the Golgi, and it's going to get to the cell membrane, and it's going to be on the inside. And so it's kind of nice to go back to this, because there is that huge difference between the inside and outside of the membrane. And we talked about this a little bit when we talked back in chapter 16 about signal transduction. So the idea is that it's just biochemically clever to anchor RAS, because that's where it's going to do its job. So it's going to be right there, so it can get activated, and it's going to be right there. And we'll talk more and more this week about how the phospholipids and the molecules like RAS, they can diffuse really rapidly. They are randomly diffusing really rapidly, and they're staying on the appropriate side of the membrane, but they're going side to side really fast. And so that's what's showing here. We just kind of emphasize, oh, it's getting activated. But it's also showing that it's also just diffusing around really fast. And so it gets activated, and it just slides around until it bumps into a target protein and starts activating it there. And we've seen that in a couple of different examples, like that PIP3, well, that's in the membrane, and it gets phosphorylated from PIP2 to PIP3, and then it's just sliding around. So this is a good kind of point in time to emphasize that again. And stuff on one side stays on one side unless it gets help to flip around. And we have that one good example of that with the scramblases in the apoptosis pathway, where it's going to flip phosphatidylserine. And the stuff stays on one side because the phospholipid bilayer has these hydrophobic tails, has this little thin layer of hydrophobic environment, and these are all hydrophilic. And so they just can't flip without some help. They can slide around on either the inner leaflet or the outer leaflet, but they just can't flip around on their own. And that's true for any of the phospholipid bilayers in the cell. So it's true of the membrane, the rough ER, and those vesicles, and the Golgi. Whenever there's a phospholipid bilayer, the molecules are restricted to one side or the other unless they get some help. Okay, so that's just following up on that example. And here's just the only picture of it. Oh, Sophie, did you have a question?
Yeah, did you post the lecture from Thursday on Facebook?
Oh, I always try to. If you don't see it, it's just because I got interrupted. Okay. And so if you see that, just send me an email. Okay. And but I'll remember, I'll post it before I post this one today. Yeah. If I didn't.
Yeah, I didn't see it. So
Yeah. Okay, thank you. Yes. And that definitely happens, because I get going on stuff. So just it doesn't add any new information that we need to know, but just really about the only image I could find on the web showing that orientation of RAS. So here's the rough ER, and these colored molecules are showing that synthesis of the RAS and anchoring it to the membrane, and then heading to the inner leaflet, like that. And it's another nice figure, nothing we need to know for our exam other than what we already showed in previous slides, but it just shows this because there are interesting exceptions to the rule in biology, and it just shows that in humans there are 3 different members of the RAS family. We mentioned that earlier in our course, and we don't need to be aware of that, just call it RAS. But they are mutated in different forms of cancer because the different family members of RAS are prominent in different tissues. And so like scientists really want to know all about him and how, well, how's he working, what's he going there. And this just shows kind of interestingly that 2 of the family members get synthesized like I described and like we should learn. The third one actually takes a little wacky path, a unique, distinctive path to the cell membrane. And that's exciting for somebody who's studying RAS and cancer biology, but it's advanced, right? If we start out with this, it's always in biology good to learn the rule, and then when you hear an exception, don't be like, oh, that Professor Champlin was a loser, but rather say, okay, we built a foundation, because exceptions are always like, oh, nice, like that, oh, how does that work, like that. And that's true for this small number of proteins. Most proteins fit those pathways that hemoglobin, insulin, the PDGF receptor, and the lysosomal proton pump go in their process of finding where they need to get to. But there are some exceptions. This Kirsten RAS is an exception. So again, just to show you that interesting fact, okay? And so RAS would fit in this part of the list, right? It's like the PDGF receptor and the lysosomal proton pump in that it's a membrane protein. It's odd in that the reason it's a membrane protein is not because it's in the membrane, but it gets anchored to the membrane. And it gets anchored to the membrane right when it gets made, like that. Okay? Fabulous. Okay? So then I just wanted to show you this again, because we plowed through this pretty quickly. We plowed through the role of post-translational modifications, and there's a lot of that in biochemistry. And this is what I just made another slide to show you this, because I had this image from a review, and it would be good just to turn it into a list, right? Because that's what we want to get out of it. We want to get the potential to say, list some post-translational modifications that are important in muscle differentiation, muscle health, right? So I picked muscle because we learned about it early in the course. We're like, oh yeah, muscles get made, like that. It's gene expression, we've got to turn on the genes. Well, there's always more to it than that. And this is not exceptions, it's just additions. And so it's a good example to go back to and say, oh yeah, there's, in addition to regulation of gene expression at the level of transcription, there's regulation of these proteins important for muscle differentiation and muscle health, even when they're made and done being made. And so I just turned it in. We talked about this, and I just turned it into a more visible list, because that's what we really want to get out of it. And with the ubiquitination, that's the only one we kind of dove into a little bit. Because when people say ubiquitination, that ubiquitination is adding a covalent ubiquitin molecule to a target protein, and it almost always involves targeting that protein for destruction. That's the most common way that proteins get degraded in a cell. So the proteins that we talked in our course about, oh, they're going to get degraded, we didn't bring it up, but almost always they get destroyed because they get tagged by ubiquitination. It's like the ubiquitin is a little flag that's, that's delivering me into this proteasome. And I highlighted that proteasome because that's a little structure in cells of many. There are many, many proteasomes in every one of our cells, and it's just involved in chewing up proteins. And so it's like a compost bin for proteins, and it releases their individual amino acids. So great. And that fit into the context of, post-translational regulation of muscle growth, muscle aging, and sarcopenia. Like that, great. So then we got to this point, and I just want to show you this again as we roll into chapter 14. When these vesicles form, they're typically just called, oh, it's a vesicle. And a vesicle is just a little phospholipid coated or surrounded sphere. And this just shows a common way that they are formed by budding. So we learned about that some. I don't know if I mentioned that it requires GTP, so it requires some energy to pinch off the vesicles. So a regulated event. And also, did I get this on here? Oh yeah, so I added this from last class because I talked about this last time, and we'll have a good example of clathrin coming up in this chapter. And so clathrin is coating that vesicle, and it actually helps the vesicle to form the bubble. And then that dynamine pitches it off. So a nice little story. Because we always start out saying, oh, you know, it just pinches off like a little soap bubble. But it's a regulated process to create it and form it. And then we talked about, and then there are regulated processes to join that vesicle with the target. So we could get a vesicle pinching off from the rough ER, and it has newly synthesized things in it, like maybe insulin in the lumen, and maybe RAS on the surface, maybe a PDGF receptor in the membrane, and it's going to get delivered to the Golgi, where it's going to fuse like that. And so just an example of some famous proteins involved in regulating that fusion, bringing those together, because that process is very regulated, okay? And then, let's see. I'm going to skip ahead because, I added some slides. Yeah, because that pinching together, or that fusion and that budding have special terms at the cell membrane. And that's the last thing we talked about last Thursday. So here's exocytosis. And it's a kind of fusion, and it requires those snare proteins and other proteins that play that role. But it has this very important special term, exocytosis. And so that's the last step in the synthesis of insulin in our pancreas, is to release it by exocytosis. So the green would be insulin or another secreted molecule like that, like cell signaling molecules like Wnt or PDGF, so forth like that, lots and lots of stuff. Or delivering proteins like PDGF to the cell membrane, like that, or RAS to the cell membrane. You would just draw a different color dot. The PDGF would be like a blue dot in the membrane. The RAS would be a red dot on the outside, and you deliver it to the membrane by fusion with the membrane of a vesicle. That's called exocytosis. And then endocytosis is the opposite. It's budding into the cell, new vesicles from the cell membrane. And there are three types of endocytosis, especially. One is phagocytosis, pinocytosis, and we're going to talk about an example called receptor-mediated endocytosis today. So I added, I guess, a couple of things in there that got those separated from when we talked about last time. So then, as we're rolling into chapter 14, another thing is that in chapter 14, the book talks a lot about the phospholipid molecules and other hydrophobic molecules in cell membranes. It shows a lot of structures to them and things like that. And we don't need to learn those structures. And in general, we don't need to learn those names, no matter what. You don't have to go, my gosh, I'm going to memorize a lot of this structural stuff or name stuff from the tables and stuff in the chapter. I wanted to remind you that we did learn, and we need to know the names of 2 phospholipids so far in our course, and be able to, include those, not in our third exam, but certainly in our final, is to bring up examples where we bring together our knowledge of the cell membrane and back to that role of phosphatidylserine, where it's synthesized exclusively present on the inner leaflet of the cell membrane. And of course, it would have been synthesized in the smooth ER and traveled through vesicles to the cell membrane, and then it gets flipped. It gets on the outer leaflet using this enzyme scramblase. So we know about that one. It's in the book in chapter 14. It shows the structure of it. Don't need to know the structure of it. Just need to know, it's played a role in apoptosis. It's in the cell membrane. And then this other one we learned about in chapter 13. Phosphatidylinositol, we like to call it PIP3, because it has 3 phosphates on it when it's activated by PI3 kinase, and it's involved in cell signaling and signal transduction, like that. And so again, here's the structure of phosphatidylserine from the book. Don't need to know that. It is good to appreciate this classic drawing, right, where we say, from intro bio, we say, well, that's the hydrophilic head. and then it has these two fatty acid hydrophobic tails. And that's the level of, detail of structures that we need to know from that, okay? And the book also has, here's that phosphatidylinositol, or PIP, that we learned about in signal transduction. Same idea, hydrophilic head and hydrophobic tails. They're called phospholipids because they have a phosphate group and these lipid tails, these very hydrophobic tails, like that, okay? And I just wanted to show you this, just to go back and appreciate the naming of these from that signal transduction chapter, because now that we're focused more on the membrane, we can look at this a little bit. So I don't think there's anything new here that you have to learn, but just seeing it now that we're talking about that. And so this would be the synthesis of the PIP2 and in the cell membrane. And so there would be enzymes that would be catalyzing it. Those enzymes would be proteins that were synthesized by free ribosomes and just released into the cytoplasm, and they're just cruising around ready to phosphorylate stuff. And it includes that PI3 kinase that phosphorylates PIP2 to make PIP3. And here it's just showing, why is it called PIP? Because it's got initially phosphatidylinositol, and then it gets a phosphate group. So that's PIP, and then another phosphate group, that's PIP2, and then a phosphate group, that's PIP3. And you can see that in this case, right, the organic chemists and biochemists, they're just listing the positions of where the phosphates get added on that carbon ring. So you're familiar with that. We don't need to add that, just PIP2 is made by PIP3. What we should do is see this and be able to say, and just remind us again for the final, this idea that this chemical reaction is catalyzed by that PI3 kinase. And this reaction is catalyzed by phospholipase C. And you might not remember today, but we learned 3 different signal transduction pathways for receptor tyrosine kinases for our second exam. One involved that RAS cascade pathway. Another was this PI3 kinase. That's the one that was going to regulate the FOX out. And then one that wasn't in our book anymore was this phospholipase C, like that. And both of these enzymes use this PIP2 as a substrate. And this one makes PIP3, and this one cleaves it, phospholipase cleaves it, and we get the DAG out of it, that's this. And then we get this out of it, and DAG stays in the membrane because it's the hydrophobic tails, and this PIP goes out into the cytoplasm, and it's called PIP3. And that's kind of interesting because, right, just to kind of dive into that a little bit, because now we're talking about these molecules a little bit more, is because what happens is, oh, this is PIP2, and this enzyme is going to add a phosphate, so we get a PIP3. This one is just going to cut PIP2, so how do we get PIP3 out of it? And the answer is it cuts it here. So what floats away is a PIP3, and we don't have to distinguish it, but that goes back to the naming, right? There would be different numbers here to distinguish this PIP3 from that PIP3 molecule, like that. Actually, this is called IP3, sorry, typo by me, right? And naming by me, IP3, IP3, right? But it's a 3. Why is it a 3? Because it actually picks that one up. So, just a little bit of a detail, and not a really important one for us to be able to recreate on the exam, but just a way to say, yeah, no, we're gonna remember those phospholipids and not worry about the others, and not even worry about the structural details of these for exam 3, like that. Okay. So we learned about those guys, right? And so we've learned some about this pathway, and that's talked about in more detail in the chapter 14, because things are heading to the cell membrane. And the cell membrane components get made either by the rough ER or the smooth ER. The smooth ER is going to be making the phospholipids and the other lipids and fats that are in the membranes. So that's going to come up again. And at the cell membrane, we're used to thinking about it as like, oh, it's just doing its job. The cell membrane has got really interesting traits, characteristics. One of them is that when a cell grows, any cell on our planet, any cell on our planet grows, the cell membrane has to get bigger. And so there's always this dynamic balance of endocytosis and exocytosis. And we think of examples of, well, we got to exocytose this vesicle so insulin can get released. When we do that, we're actually adding membrane to the cell membrane, right? Because the vesicle fuses with the cell membrane and adds its phospholipids. And so there's always this dynamic process of endocytosis and exocytosis, keeping the membrane to be the size needed. And if the cell is growing, there must be more exocytosis than endocytosis. We must be bringing new molecules of phospholipids to the membrane. And in examples that we want to focus on in chapter 14, they include this idea of this membrane traffic and protein traffic that we've already learned about, and how that balance is occurring and what's happening in comparing exocytosis and endocytosis. And that's why we kind of wanted to super underscore those terms. Endocytosis would be budding into the cell. Exocytosis would be fusing vesicles at the cell membrane like that. So lots and lots of that. And here's, again, we talk about names and history. So in 2013, so 13 years ago, 3 scientists won the Nobel Prize in Medicine for studying this membrane traffic, protein traffic. One of them discovered those snares, for example, like that, and they're just like, oh my goodness, such an important thing in normal health and all kinds of, diseases and health problems like that, right? And so again, don't need to be able to read this, but it's just interesting to see this, right? 16 years ago, these scientists did most of their research kind of in the 1980s and the 1990s especially, so 25 years ago was when all this was getting done, really set up. And it's just cool to see. One of them studied membrane traffic in yeast, because yeast are single-celled eukaryotes, and they are closely related to us, like that. You can easily find orthologs of cell membrane traffic regulators in yeast and every other eukaryote. Every plant and every animal has this process, these processes going on. So one studied yeast and really studied the genetics of it, looking for mutants. We have defects in the yeast, and mutations in genes that encode proteins that are like the snare proteins and so forth, the clathrin protein and so forth. And then this is the scientist that discovered snares, and we'll talk a little bit more about him, because what he was really doing is he was studying heart disease. And snares play a role in heart disease in normal health, like that. So we'll dive into an example of that. And then this scientist studied that process in neurotransmission. So again, only a few of you took neurobiology, but we use another example where we talk a little bit about anytime we're thinking, our nerve cells, our neurons are releasing neurotransmitters. And neurotransmitters are present in vesicles in the neuron, and they get released by exocytosis. And we're thinking so fast, we need to recycle that membrane and those neurotransmitters. So at the step of where we have 2 nerve cells talking to each other using neurotransmitters, there's just a tremendous amount of this vesicle traffic of exocytosis and endocytosis, just like that. Because we think a lot and we think really fast. So really great examples of people who have studied this topic like that. So in this chapter, it's one of these things where, in addition to the idea of recalling like pictures and drawings, it's good to recall stories and fit these topics into a story. Like I remember when we learned about this in college, we learned a little bit about heart disease in college. And so we learned about something there, like that. And so what I'm going to do in lecture, and our book really, it talks about this, but it talks about it in a more of a kind of a flowing, just introduce this happens, and this happens, and this happens. And I just think it's better to kind of reorganize it into some memorable examples. And we'll talk about them one at a time, and it'll chew up, the rest of the day and most of Thursday, because it's going to be our way of getting through almost all of chapter 14. So 3 examples, and they all have relevance for us walking around. And, just to remind you once again, as a molecular biologist, I just don't care about that, I just love the nuts and bolts things. Like that RAS, oh man, it's anchored in the membrane. I'm just like, geez, man, oh gosh, that could be such a cool tattoo, like that. And but so here they are, and again, we'll walk through them one at a time, like that. And you can see I've added things like urine production and heart and stuff like that, to help provide a kind of relevance to it that will fit it into. But ultimately, we're not learning about, human anatomy and physiology. We're dropping down to the molecular and cellular level. So here's the first one. So there's a protein called aquaporin, and it's a cell membrane protein, and it looks like a donut. And water goes through the donut hole. And it goes in and out, and it's not pumped or gated or regulated in any way. It just lets it flow through. So what's the function of aquaporin? It just increases the rate of osmosis. That's it, ever. Well, I should mention that aquaporin is a gene family in us, and it has evolved. Some of the family members do let some other small molecules in and out of cells. But primarily, aquaporin has a great name, aquapore. It's just literally a channel. And you learned in introbio that osmosis is water moving across the cell membrane. Sometimes the osmotic concentration is the same on inside and out, so this situation is isotonic or low concentration of solids outside, hypotonic, hypertonic. In introbio, it's usually just left at, the cell membrane is leaky and water can diffuse in and out. It is leaky, but in no cell can that leakiness be fast enough. The osmosis does not occur fast enough. And in every one of our cells, we have tens of thousands of aquaporin donuts in our cell membrane to allow osmosis to occur fast enough for life to occur. And then there are specialized situations where the osmosis rate needs to be super high. So when we cry, the water in our tears is coming out of aquaporin channels. Or when we sweat, or when we produce urine. It's all about water and passing through cell membranes fast because of aquaporin. And aquaporin doesn't change shape. So I talk about it in my intro bio course because it's such a great protein, because it doesn't change shape. It's just a big protein donut, and it's got a channel, and the channel is just the right size, and it's a physical hole like that, just the right size for water. And then also, water is super hydrophilic, and water wants to make these non-covalent interactions, these hydrogen bonds, either with water molecules or other atoms. And the channel has just perfectly spaced amino acid side chains. So as the water goes through, it has to touch those like that. And so it's a physical and a chemical channel. And the water is going in and out really fast. And it's great because, in molecular biology, we can talk about the physical features of molecules, structural, spatial features, and you're like, I got that. But we can't possibly comprehend is time and rate in molecular biology. Because through one of these channels, every second, several billion water molecules pass. And they have to make those hydrophilic, non-covalent interactions as they pass through. That's how weak those interactions are. You know, you learned about them, right? They are why water has surface tension and all its wonderful qualities. Those interactions between water molecules, they're called hydrogen bonds, and it's given a special name. They are very, very weak, and they don't last long, right? Less than a billionth of a second, like that, right? And the water's just going in and out, in and out, in and out, and it's just going in and out either way. And it's that many. It's like, imagine the entire human race passing in and out of that door every second, constantly, you know? And getting carded, right? You get, you know, you have to, it's a chemical filter too, right? Like that. Just, you know, and you're just like, man, you know. And what's the next thing to say? Every eukaryotic cell on our planet has orthologs of aquaporin. And that's because it initially evolved in bacteria. So lots and lots of bacteria even have aquaporin. And it's just so important for osmosis, right? So that's our first example. And we could point to tears or sweat, or we're going to point to urine to show how it plays its role. And it's just going to be osmosis. So yes, orthologs of aquaporin present in essentially all eukaryotic organisms. You know, some cells give it up for some special odd reason, you know, like that. But very ancient and just classic, like, you know, where people are like, I don't know about evolution. I'm like, me neither. It's pretty awesome, you know, like that. It's like crazy awesome. All right, so here's the kidney, right? And again, this is not going to be on your test. This is, you know, anatomy and physiology. Our kidneys filter out wastes. And so here comes the blood, and it's going to filter this out, and this yellow is going to become urine. And so that's called the glomerulus, and you know, if you took human AMP, you learned about the kidney and the glomerulus. That's the little filtering unit. Each of our kidneys has about 500,000 of these little filtering units, and that's what it does. It filters out waste. And so this is the first filtering process. Wastes are coming out, and in getting the waste out, there's a lot of water that comes out of the cell also. This first step in filtering out waste produces very dilute urine, like 50 gallons a day. And you know, the big focus is, oh, we're going to filter out the waste. We did it. But there's a second part of the story, and that is we have to get almost all that water back. Right? Nobody, you know, produces 50 gallons of urine a day. And it's regulated. Right? If you don't drink water, you don't produce as much urine. There's a regulation of how much of it gets brought back. And who's in charge? Aquaporin. Because you can get it, you know, coming across them, remember, like that. So this just shows that there's the filtering unit, and there it's produced, and then here's this long tube, and this is where we're focused. This is where we're getting water back into the cells and adjusting it based on how much water the organism needs. So our brain produces hormones that go to the kidney that say, retain more water. Produce a smaller volume of urine. Or you're chugging water, get rid of the water, right? So that's regulated. So we're going to see that. We're going to see that here is our exclusive focus, is the waste has been filtered out. The waste is in this pre-urine liquid, extracellular liquid, and we need to get a bunch of water back. And it's going to come through aquaporin channels in the membrane. So this just shows a more, you know, kind of drawing of it. Here's that first filtering, very, very dilute urine. And here's our focus, is the blood vessels, again, get close here, because they're like, now we have to get lots of, like almost all the water back. Leave the waste molecules in the urine, but make them way more concentrated. And what we're going to do, it's not about anything about the waste molecules, they're there, it's get the water back. Aquaporin is great for that because it's exclusively going to let water come back. It's not going to let any of the waste molecules come back. It's a really great filter. And so here's just a zoomed in drawing of a cell. And on one side of it, here's that very dilute urine. And on the other side, here's the blood. And there are aquaporin channels here, and they're getting they're getting water back in, like that. And they're getting water back in by pumping it. They never pump it. There just has to be setups of having different concentrations. And they don't set that up. They just are a passive filter, like that, okay? So what we see here is there's aquaporin on the cell membrane, like that. And there's aquaporin on this cell membrane too, because we gotta get it over here. Not by pumping it through aquaporin channels. They're just a passive chemical, physical filter for water through. And it goes in both directions. Here, the arrows are this way just to show that primarily it's coming in and going out, like that. And they are listing here, again, we don't need to know anything about the details, but it's a gene family. The gene family is like 10 members. In humans, they just are numbered. And you see there's some different roles. They're the same protein, same donuts, just slightly different, and where they get expressed in the body and where they get located. Remember we talked last time about how traffic has to move differently, right? Oh, we're taking to this cell membrane. Well, here's a situation where that cell membrane is different from that. So we got to make aquaporin and deliver it to the appropriate side of the cell, like that. So it's just a really good example, again, to set it in your mind of, I'll never forget that because it's how I produce urine that's concentrated and how I regulate the volume of urine, okay? So this is going to show, here's the exclusive part of our story is we can adjust how much water gets pulled back by how many aquaporin molecules are in the cell membrane. And we do that by having extra aquaporin in vesicles and add it by exocytosis. So this is an example of dynamic balance, is we adjust how much water comes back into the body by adjusting how many aquaporin molecules are in the membrane. And so here's a hormone, don't need to know the name of it, but you would have learned it in human ANP if you took it. It's called vasopressin. It's made by the brain, and it's when you're thirsty and you want to retain water and reduce the volume of urine. And vasopressin comes through the blood, a hormone, binds to a receptor, and signals this cell to put more aquaporin onto the cell membrane. It's just a very, very satisfying visual, right? Because we don't have to make new aquaporin, we have it, and we just need to put it on the membrane. Is this aquaporin helping? No, but this aquaporin is. If we put more there, we'll get more help. And so that's what vasopressin stimulates, is exocytosis. By stimulating vesicles containing, I didn't even get that word on there, aquaporin, to fuse, to go through exocytosis. Is anything being released? No. This example is just adding or subtracting aquaporin from the membrane, right? Because if you chug a bunch of water, you're going to be pulling aquaporin off the membrane, and you let that urine go. There's way too much liquid in me, like that, and it's regulated just by that, like that, right? Like that, right? Just add it, and you can see that the whole vesicle, the phospholipids get added, right? It's going to be a dynamic balance, right? Of, elsewhere we're going to have to be pulling some membrane back, but leaving the aquaporin like that. So it's all regulated, but in a very simple way. And we just want to focus in on this as an example. And that's it. That's all our whole point, is to zoom in on that as an example to see that dynamic balance of that regulation like that. So pinching vesicles containing aquaporin off the cell surface leads to an increase in urine volume, right? And that's just going to be a 1 or 2 point question on our third exam or our final, right? Of what do we learn about aquaporin? It regulates osmosis, and it plays a big role in every cell on Earth, not on Earth, every eukaryotic cell on Earth, just doing that. And then, once you have that, you can evolve important physiological roles like that. So that's our good example of that, and a good example of this dynamic balance of exocytosis and endocytosis Great. So that's our first example, and it's just a really nice package. And now our next example is about brains, and very, very similar story of exocytosis and endocytosis, like that. And this is a pretty quick one, but it's cool to see kidney, brain role for these. And so just, again, not for our exam, but just to set the context for everybody to appreciate it, because only a few of you took neurobiology. Here's two neurons. Neurons are nerve cells that do all our thinking and moving and feeling, and they're connected by a synapse. And a synapse is where these two cells get really, really close together, but they're not touching. And this neuron is the first in line, and it's going to send an action potential signal down here, and then that action potential signal is going to continue in this postsynaptic neuron. And this little red connection is what we're going to zoom in on to look at that. And so here's the green presynaptic neuron. It gets close to the postsynaptic one, but it's not touching it. And it needs to send that thinking signal, that electrical signal, that action potential. It needs to jump over that and get it going in the postsynaptic neuron. And the way that happens is by releasing molecules that are called neurotransmitters. And see them there? They're in vesicles, and they get released by exocytosis. And they are signaling molecules of the type we talked about. They float, diffuse across this very short distance, and bind to receptors on the postsynaptic side. And those receptors are little ion channels that open and close in order to get that action potential going on the postsynaptic side. For us, for our course, our example, all we're doing is we're zooming into this location. And what we can say right now is in our brains, every second we're alive, there's just an astonishing amount of exocytosis happening. Remember, and that's going to be those snare proteins, helping to get those membranes together and fusing them. And it's called exocytosis, but we're fusing it like that, right? And yeah, because that happens every second, many times a second, like that, over and over again, okay? So that's the first vision. These neurotransmitters are little molecules that get synthesized in this green neuron, and I don't think any of them. Well, I don't think any are. None of them are proteins. Some are peptides, I guess, but they tend to be just little tiny organic molecules, and so that's synthesizing it and releasing it. Then here's the next thing. You can't just let those hang around there. You want to have them there and gone. And the way that happens is by taking them back up into the presynaptic neuron by endocytosis. So I think that's the next figure, like that. So they're doing that. A bunch of them get released, and then get taken up again. That allows these to be recycled. And it's the neurotransmitters that are recycled, and what else? The phospholipids, right? Because we're adding tremendous amounts of phospholipids to the cell membrane, and then we're pulling them back in. And just one example is this is a famous neurotransmitter called dopamine, and here is a dopamine receptor that binds it and pulls it back in. In this case, the dopamine is not coming back into the vesicle, it's coming back through a special channel. But in other cases, the vesicles are capturing the neurotransmitter and pulling them in that way too. In any case, we have to have lots of endocytosis to balance the exocytosis. So it's this dynamic balance, and but it's this pathway, this recycling over and over again pathway is happening over and over again. We do make new neurotransmitters because they do get used up, broken down, and things like that. But mostly our thinking is about just recycling them, bring them back in, so, send an action potential on, and I'm not a neurobiologist, but like when a nerve cell's firing, the rate that it's sending an action potential could be thousands of times a second, I think. So that means we're going to let it go and then get it back in there thousands of times a second. So it's another example of, well, we can't possibly imagine the time, how fast this is happening in time. We can draw out the spatial aspects of it and appreciate that. And so that cycling is just a huge part of the thinking process. And it's regulated. And you can't see the details of this. That's fine, because we're not here to learn that. But just to add relevance to this is many, many medicines and illegal drugs work by adjusting the recycling process, slowing down the uptake increases just like right? And dopamine is one of the molecules like endorphins that makes a person happy like that and excited. And these boxes are just describing drugs, legal or illegal, that speed up or slow down these processes. So inhibition of reuptake, cocaine, amphetamine, benztropine. And again, those are names not going to come up on our exam, but just over here we have amphetamines and perfenazines and stuff like that. And then what are they doing? Interference with vesicular storage, stimulation of release, blocking of dopamine receptors, and so forth, like that. So just a lot of relevance for our health happening there, and just happening in that cycle. And that's the message there. The message for us is it's very similar to the urine story. Lots of excitosis, lots of endocytosis, very regulated, just like that, very adjustable process. Great. This is just another slide, just to try to give you another visual of that. These neurotransmitters are getting recycled, released by exocytosis, brought back into the cell by endocytosis, like that, and as well as the membranes, like that. So that's that one, good one. Then the next one, it takes a little while to talk through, and it's this receptor-mediated endocytosis and a special relevance in heart disease. So I mentioned that, 13 years ago, those Nobel Prize winners got it for membrane traffic. And one of them discovered snares. He was studying heart disease and how that it becomes a problem in some people, like that. And the process we're going to turn our attention to is called receptor-mediated endocytosis. So we already learned that term, and let me see if I can find it. Might be best just to scroll back to that term. I didn't get it copied in here. It's a little bit of scrolling, but just to show you that again, right? So we have that exocytosis term we've been using, and then endocytosis. So phagocytosis, really the common one where you're going to engulf something like that. Pinocytosis is often called cell drinking because you're just bringing soluble molecules in. And then here's our example. It's a type of endocytosis, and it's called receptor-mediated endocytosis. And this is a good figure to compare and contrast, because you can just see this. So here, you're not really bringing these molecules in with any real selection or concentration preference. You're just like go, like that. Like that, like when you're drinking, you're just drinking whatever the molecules are in the glass. Receptor-mediated endocytosis is where you're trying to pull in specific molecules, and so that's what the receptor part is. We have a receptor for these stars, and we're going to bring those stars in. And the stars are soluble molecules in the liquid that are not particularly concentrated. So pinocytosis is not going to get it done. And the receptors are these green molecules. They're transmembrane proteins on the surface. We've talked about receptors. These are not receptors for signal transduction. They're receptors to grab, like a hand, and then have the molecules get pulled in because the receptors get pulled in. And so something to notice here is that we would have these receptors just on the surface of the cell. And then a key concept is that when the receptor binds the preferred molecule, the ligand, that's what we want to bring into the cell. So a key concept in receptor-mediated endocytosis is the cell is able to distinguish between the ligand-bound receptor and the unbound receptors. So see that? We got scattered receptors that haven't bound to ligand. And then when they bind to ligand, those are the ones get pulled in. And it's a real satisfying cell biology concept to see that happening, right? We have to have a cell to see what's going on. And what's going on is the ligand-bound receptors start to cluster together on the cell membrane. That's not showing well here, but that's what's happening. So there's somehow, when the ligand is bound, those receptors can be identified and brought together, and then that's how we get a concentrated amount coming into the cell through this endocytosis process. So another way to think about it is receptor-mediated endocytosis is fancy pinocytosis, specific pinocytosis, right? We're bringing liquid in, but we want to increase the concentration of specific molecules. The way to do that is have receptors, and when the receptor binds, have them start getting clustered, okay? So we'll just add some terms here, because who's going to help them cluster is this red molecule, and that's this protein clathrin. So we already learned that term, clathrin, and I'll just, since we're close to it, I'll just scroll back to that slide. Right, we saw that. There's that clathrin-coated vesicle. And so this actually would be a general drawing, and then clathrin can play this specific role, and we'll talk about that. And so let me go to this and draw out a little bit about what we see here. And so we can have this, and we'll just show it like that, okay? And so here's the cell membrane, and we can have these receptors in them. And let's start naming the receptor. The one we're going to learn about is called the LDL receptor. And the molecule that it binds is a big molecule, a particle called LDL, low density lipid protein. And we'll dive into the details. LDL is carrying cholesterol. So we'll look at drawings and images of what that's like. And so we're going to be bringing cholesterol to the cell. And every cell needs cholesterol, so we'll learn a little bit about that. And it wants to bring the LDL into the cell. And if you've heard a little bit about LDL receptor, we'll also learn it's called bad cholesterol, and we'll learn about why, like that, okay? So here we have these receptors, and then there'll be some of the receptors that get LDL bound. And when they do, they start to cluster. So let's just draw what that would look like. So here's one that just bound it, hasn't clustered yet. But these have clustered, and why do they cluster? When they bind, the shape of the LDL receptor changes. So it looks a little bit like a pair of pliers, and so the pair of pliers has like a pivot, and when it closes on something to pinch it, the hand grip is also changing its shape like that. So it's a little bit like a pair of pliers. When it grabs it, the position of the handles change. And so you could draw it like that. So these guys all have a little bit different shape. And just to, to zoom out and remind you that although these aren't receptors, these aren't going to be signal transduction stories. This would have, the transmembrane ligand binding, the extracellular ligand binding domain. And then it would have a transmembrane domain that crosses the membrane. And then it would have a intracellular domain that would be on the inside. And the protein is set up to be like kind of pliers, where when the extracellular domain changes shape, the intracellular domain changes shape as well. And the next thing to mention about that is just to add it to our list. Where do we first see newly synthesized LDL receptor? Well, the membrane of the rough ER, right? It gets synthesized just like the PDGF receptor. It's a transmembrane protein, and it needs to get synthesized in the rough ER. Then it goes through traffic to get to the cell membrane like that. So here it is on the cell membrane doing its job. And so you can see that the bound ones have changed shape, and that's how they get clustered, because the clathrin protein is actually a trimeric protein. So here's some clathrin protein to draw that out, and this is a certain form of clathrin. And it's a trimer, and it has that type of shape. Each of the monomers has a binding site, and it can only bind to the intracellular domain of LDL receptor. So see, it's not bound here, but this one's bound, and it's ready to bind others. And that's how the receptors get clustered, is because the clathrin is a trimer, and so it can hold 3. And also because it's a trimer, we can have clusters of clathrin, because this one could be bound here and bound to another, ready to bind to another one and bound to this one, right? And so that's the actual physical basis for the clustering, is that the receptor is scattered across the cell membrane, and its intracellular domain is a shape that doesn't bind to clathrin, and then it binds to LDL, and that changes its intracellular domain. Clathrin binds it, and clathrin is a trimer, so it can hold 3 of them. And it can create networks of binding, because more than one clathrin can bind to each one. And so this is where they cluster, and you can see that when we talked about clathrin before, we talked about it being a coated vesicle, because under the electron microscope, the scientists can actually see some fuzziness here. And so those scientists, back in the 1980s and 19s, they called this a coated pit, because there's a little dent in the cell membrane that has this little fuzziness to it. And the reason it forms a dent is also what I tried to show here, and that is that clathrin has a shape to it that causes the receptors to kind of start to form that dent, like that. And so the coating clusters the receptors. The coating also starts to form a little dent, and then a bigger and bigger and deeper dent, so that we start with a coated pit. And I'll just erase this, and I might not get a chance to use all the right colors, but what's going to form then, I guess I'll be using, I don't remember what I used, but what's going to happen then is we're going to get a lot of these ligand-bound receptors clustering, and the more they cluster, the bigger the dent, deeper the dent gets, the bigger the dent, because of the shape of the clathrin trimer. Because the clathrin trimer forces these to be held in this kind of triangular kind of bubble-like shape that forces it to form more and more of a dent. So we get more and more clathrin, and we get this coated pit, and that's going to pinch off through endocytosis, right? We're going to have endocytosis occur, and we're going to make a coated vesicle, because we're going to get to this, and it's going to pinch off. It's going to form a fully separate bubble like that, and it's going to be coated with clathrin, because that's what gave it that spherical shape, first the dent and then the shape, and we have a coated vesicle. And what's happening is through this endocytosis, we're bringing the LDL into the cell. So this cell needs some cholesterol for its purposes, and it's bringing it in. The cholesterol is here in the blood, and we're bringing in a concentrated amount of cholesterol into the cell, because the LDL receptor is grabbing these cholesterol molecules. They're part of this LDL particle, and we're getting them into the cell. And so that's receptor mediated, right? It's endocytosis, but it's bringing in, by preference, cholesterol that's in LDL particles using the LDL receptor. So it's a kind of endocytosis. It's happening at the cell membrane, right? We're bringing it into the cell, and it's a type of budding, so it's going to use that dynamin to pinch it off at the end, but it's really that clathrin that's helping to form the shape. And clathrin has that because of its special trimeric nature, like that. So that shows you how the receptors, when they bind the ligand, they start to get clustered, and then the clustering also causes the dent, the coated pit, and then it pinches off, like that. And so classic cell biology, because these people first saw that happening under electron microscope, and they're like, what's that coated pit, coated vesicle thing? We'll talk Thursday about the next step. The next step is to uncoat the vesicle, and then send the vesicle to where? Send it to the smooth ER, because that's where cholesterol gets dealt with. And so we'll talk about that. And then the clathrin goes back to the cell surface, and so there's a cycle of clathrin helping form coated pits and coated vesicles, then uncoating those and bringing the clathrin back to the cell surface like that. So it's a really good example. And again, we'll see it's all about heart disease, because bad cholesterol is bad, and should be getting it out of the blood, blah, blah, blah. So we'll learn a little bit about that, some of that physiology, and why the scientist that, won the Nobel Prize, was involved in that kind of research, like that. There were others too. That guy was in Connecticut. The other heavy-duty people were in Texas, collaborating like that. Okay, great. So we got to stop here. But in doing these examples, we're doing a great job, getting examples from chapter 14. Awesome.